Hello guys, welcome back to Biology 360 YouTube channel. Today we are going to see Chapter 3 Plant Kingdom from the unit Diversity in the Living World. We know that R.H. Whittaker gave 5 kingdom classification. In lesson 2, we have seen first 3 kingdom, that is Monera, Protista and fungi. In this lesson, we will continue with 4th kingdom, Plantae. Check on the description for second lesson and first lesson. Let's start. So here we are going to focus on the 4th kingdom, that is plant kingdom. The classification bases have been changed. Earlier we kept the organism that has cell wall into the kingdom Plantae. So fungi and members of Monera and Protista having cell wall were kept in Plantae. But now we have excluded it from Plantae and we have given separate kingdom for it. In this chapter, we will describe Plantae under the topics algae, bryophytes, pteridophytes, gymnosperm and angiosperm. Okay. In this paragraph, we are going to see different system of classification. The first one was artificial system of classification that was given by Linnaeus, and it was based on morphological, vegetative and sexual characters. So you see, vegetative characters and sexual characters were given equal weightage. That was the disadvantage of artificial system of classification, because vegetative characters like the number of leaves, the shapes of the leaf, the color of the leaf are easily affected by environment. How could we take that into consideration? So this was the disadvantage of artificial system of classification. And the second one was natural system of classification that was given by George Bentham and Joseph Dalton Hooker. Here they classified organism not only based on the external feature but also on the internal features like ultrastructure, anatomy, embryology and phytochemistry. And it was given for flowering plants by George Bentham and Joseph Dalton Hooker. And at present we have third kingdom of classification, that is phylogenetic classification system, and it was given by Engler and Prandtl. Just make a note, it was given by Engler and Prandtl. So it was based on evolutionary relationship. It states that the organism belonging to the same taxa have a common ancestor. We always say that humans evolved from monkey, but actually we human and monkey we have common ancestor and we are not evolved from monkey. We both are present in the same taxa and we have a common ancestor. That is the truth. So phylogenetic classification explains this. It was based on evolutionary relationship. Next comes numerical taxonomy. It was based on observable characters. It gave number and codes to all the characters and it gave equal importance to all the characters. And next come cytotaxonomy. It was based on cytological information like chromosome number, chromosome structure and chromosome behavior. Next, chemotaxonomy. Chemo, it was chemical, right? So chemotaxonomy was based on chemical constituents of the plant. What are the chemical constituent it has? Based on that, they resolved and they gave chemotaxonomy. So in this, first we will see algae. Algae are chlorophyll bearing, simple, thalloid, autotrophic and largely aquatic. So they come under Plantae. So they will have chlorophyll. They are simple. What is thalloid? Thalloid means they are multicellular organism, but they does not have specialized structures like roots, stem and leaf. It is very different from what we see around our environment. We see plants that has leaf, stem and root. But these algae, they don't have true roots, true leaf and true stem. They have root like, stem like and leaf like structures only. And many of them are found in aquatic habitats and they are also found on woods, soil, moist stones and they may be in association with fungi, that is lichen, or they may be on animals like sloth bear. Next, the size of the algae range from unicellular to multicellular. So, unicellular algae is Chlamydomonas, and colonial form is Volvox, and filamentous forms like Ulothrix and Spirogyra. So, in algae, we have unicellular to multicellular, that may be colonial, that may be filamentous, like that. So marine forms such as kelps form massive plant body. So kelps is a marine form and it is of great huge size. Okay. And what about the reproduction of algae? So the reproduction of algae is of three types: vegetative, asexual, and sexual. Vegetative reproduction is by fragmentation, and asexual reproduction is by spores. The common spores is zoospores. They are flagellated, that is, they are motile, they can move. And sexual reproduction takes place through fusion of two gametes. So, based on the gametes, we categorize them into three types. That is, first one, the gametes are flagellated, that is, they can move, and similar in size, that is Chlamydomonas. They are non-flagellated, they will not move, but they are similar in size, that is Spirogyra. The main thing is they are similar in size. So the gametes which are similar in size and they fuse to give the algae, this process is known as isogamous. And if the gametes are dissimilar in size, they are different, one is big, one is small, and they fuse to give an organism, that gametes are known as anisogamous, and the example is Chlamydomonas. And if we come to the third type, the female gamete will be big, that will be large, and it will not move, and the male gamete will be small and it is motile. They fuse to give a new organism. This type of gametes are known as oogamous, and the examples are volvox and fucus. So reproduction is by vegetative, asexual, and sexual type. Next. Okay, this paragraph explains the uses of the algae. First one is, at least half of the total carbon dioxide fixation on earth is carried out by algae. So they are plants. Plants usually take in carbon dioxide and release oxygen. So at least half of the carbon dioxide fixation is by algae. And next one is, as they take in carbon dioxide and they release oxygen, they increase the level of dissolved oxygen in their immediate environment. And third one is, they are primary producer of energy rich compound. Being plants are the primary producers, right? We depend directly or indirectly on plants, so they are the primary producers. Next one is, Porphyra, Laminaria, and Sargassum are among 70 species of marine algae that are used as food. They are directly used as food. And the fifth point is, some organisms like brown algae and red algae, they have a large amount of hydrocolloids. What are hydrocolloids? They are water holding substances. They keep the water uh, in them for longer time. Example, algin in brown algae and carrageen in red algae. And the sixth point is agar. So agar is a commercial product and it is obtained from gelidium and gracilaria. This agar is used for preparation of ice creams and jellies, and it is used for growing microbes. And the seventh point is chlorella. Chlorella, a unicellular algae, is rich in protein and it is used as food supplement. Even the space travelers, they cannot take the foods to the space, so they take chlorella, that is unicellular algae, that provide them with proteins. So already we have seen a unicellular algae, that is Chlamydomonas, and now chlorella. So two unicellular algae, that are Chlamydomonas and chlorella. Okay. Now the algae is divided into three classes: Chlorophyceae, Phaeophyceae, and Rhodophyceae. We will say each one of it. First one is Chlorophyceae. Chlorophyceae is green algae. They may be unicellular, colonial, or filamentous. They are usually grass green in color. Due to the dominance of the pigments chlorophyll A and chlorophyll B. And the speciality in Chlorophyceae is they have chloroplast that is of different shapes. Like chloroplast may be of discoid shape, they may be plate like, they may be reticulate, cup shaped, spiral, or ribbon shaped. And most of the members have one to more storage bodies, that is pyrenoids. Pyrenoids are a special storage body present only in Chlorophyceae. These pyrenoids are present in chloroplast and they store proteins besides starch. And some algae are that can also store food in the form of oil droplets. And green algae usually have a rigid cell wall that is made up of outer layer pectose and an inner layer of cellulose. And if you see the vegetative reproduction, it is by fragmentation. And if we come to asexual reproduction, it is by zoospores. These zoospores are flagellated. And if it come to sexual reproduction, it is variable in all the organism. It may be isogamous, it may be anisogamous, it may be eugamous. We cannot specify it. And the examples of Chlorophyceae are Chlamydomonas, Volvox, Eulothrix, Spirogyra, and Cara. Next, we will move on to Phaeophyceae. Phaeophyceae means brown algae and they are mainly found in marine habitats. So, their size and forms are variable. It can be of simple branched filamentous form in ectocarpus to profusely branched, that is, it has more branches that are represented by kelps. We have already seen that kelps form a massive plant body and it may grow up to a height of 100 meters. So, in this, they have chlorophyll A and C, carotenoids, and xanthophylls. We have seen in Chlorophyceae we have chlorophyll A and B. Phaeophyceae have chlorophyll A and C. They vary in color from olive green to a various shades of brown. Okay, this color change is due to the presence of xanthophyll pigment, that is fucoxanthin, present in them. Due to the presence of fucoxanthin, they may be of brown color. Next, the food stored as complex carbohydrates. The food is stored in the form of carbohydrates, which may be in the form of laminarin or mannitol. This is important. The food is stored as laminarin or mannitol. So they have a cellulosic wall as usual, and outside it is a gelatinous coating of algin. Algins are what? Hydrocolloids that can hold waters. And in addition, they also have centrally located vacuole and nucleus. If you see the structure of the plant body, they have three things to note. First one is hold fast. Hold fast through which they attach to the substratum. In this picture you can see very well. And next S type, and next frond. Frond is like leaf like photosynthetic organ. We have already seen they don't have true leaves, right? So it is leaf like photosynthetic organ, that is known as frond. So three things: hold fast, stipe, and frond. Next we will move on to reproduction. Vegetative reproduction is by fragmentation. Asexual reproduction is by zoospores. Here the zoospores are biflagellate. They have two flagella. These two flagella are unequal and they are laterally attached and they are pear shaped. Okay. Next, sexual reproduction may be of isogamous, anisogamous, or eugamous. It can be of three kinds also. And this union of gametes takes place in water, and their gametes is also pear shaped, that is pyriform. Zoospore is also pyriform, that is pear shaped, and the gametes is also pear shaped. And here also they bear two laterally attached flagella. And the common examples are ectocarpus, dictyota, laminaria, sargassum, and fucus. Next we will move on to rhodophyceae. Rhodophyceae is commonly called the red algae. They are red due to the abundance of red pigment or phycoerythrin. And majority of this red algae, they are marine and they are found greater in warmer areas. More than colder areas, they are found good in warmer areas. And these are the algae that are found in greater depth. Among the three algae, green algae, brown algae and red algae are found in greater depths. Okay, and this red algae is multicellular and they have a complex body organism. The food is stored as Floridian starch, which is very similar to amylopectin and glycogen in structure. Next, we will move on to reproduction. Vegetative reproduction is by fragmentation, and sexual and asexual reproductions are by non-motile gametes and non-motile spores respectively. And if you see, sexual reproduction is oogamous. They are specific. That is, the male gamete will be small and motile and the female gamete will be large and non-motile. And the common examples are Follisphonia, Porphyra, Gracilaria and Giladium. Green algae has no complication. Brown algae and red algae confuse me a lot. So I kept a code to remember this. This also connect a topic from class 12. So I'll attach a photograph here. So this technique we will read in class 12. It is PCR and it consists of steps like denaturation, annealing and extension. So you take the first letters P, C, R, D, D for denaturation, A for annealing and for E you write F. Okay. From this you can remember red algae and green algae. How I will say. So in PCR we have R. So here we are going to tell about red algae. From this you can remember brown algae also. So R is for red algae. First one about the pigment it has. So P represent phycoerythrin. So that is found in red algae. P is for phycoerythrin. C is for carrageen. R is for red algae. D and A. So chlorophyll A and D. And F is for Floridian starch. So now we will differentiate. Okay. First, the pigment present in red algae is phycoerythrin. From P it is phycoerythrin. So in brown algae is fucoxanthin. Next, the hydrocolloid present in red algae is carrageen. So the hydrocolloid present in brown algae is algin. So R stands for red algae. Next, D and A. So red algae has chlorophyll A and D. Brown algae has chlorophyll A and C. And the storage body in red algae is Floridian starch. The storage body in brown algae is mannitol and laminarian. That's all. Through this, you can keep in mind both the algae without confusion. Now we will see the pictures and then we will move on to bryophytes. First row is examples of chlorophyceae, that is green algae. First one is Volvox, second one is Chlamydomonas and third one is Cara. Okay. Next we come across brown algae. The first one is Laminaria, second one is Fucus and third one is Dictyota. You can see that they all have a holdfast, a stipe and a frond. And last one is red algae. The first one is Porphyra and the second one is Polismonia. Now we will move on to bryophytes. Okay. Bryophytes include various mosses and liverworts that are found commonly in moist and shaded areas. Okay. And these are characteristics of bryophytes. So bryophytes are called the amphibians of plant kingdom. What are amphibians? That can live both in land and water. So here these plants grow in soil but they are dependent on water for sexual reproduction. They require a damp, a humid place to grow on. And their important role is in plant succession. That is, they help in growth of plants from a bare rock. Next, we will move on to the plant body of bryophytes. So, it is thallus, prostate, or erect and attached to the substratum by unicellular or multicellular rhizoids. What are rhizoids? So, here they have given that bryophytes are thallus. So, thallus means they lack true roots, true stem, or true leaf. They have only root like, leaf like, and stem like structure. So, rhizoids are root like structure. Here, the main plant body is haploid and it is known as gametophyte. Simply, you remember gametophyte. Gametophyte means gamete producing type. So, it will produce gamete. The male sex organ is called antheridium. They produce biflagellated anthrozoids, and the female sex organ is archegonium and it is flag shaped and produce only single egg. And these two gametes come in contact through water and they fuse to form zygote. So remember, the plant body is N, that is haploid, and they produce gametes that are N and N. They fuse to form zygote. Now it becomes 2N, that is diploid. This diploid plant body is known as sporophyte. Okay, they are multicellular and they are diploid. This diploid plant body is not free living. They will be attached with the gametophyte. Only some of the cells from sporophytes they will undergo meiosis because the main plant body is N, right? So here it is 2N. They should change to N. So they will undergo meiosis, a reduction division to form N, that is haploid plant body. Through spores, they produce spores that are N, haploid. These spores will germinate to give the plant body gametophyte. So gametophyte, gamete producing, sporophyte, spore producing. That's all. Don't confuse, okay? Bryophytes or in general have little economic importance, but they are food for herbaceous mammal, birds and other animals. And the species sphagnum, you see sphagnum is a moss and it will provide peat that is used as fuel and as packaging material for transshipment of living material. That is, they have capacity to hold water. And mosses, along with lichens, are first to colonize rocks. That is, they help in plant succession. And they form a mat-like thing in the soil so that they will prevent soil erosion. As the rain falls, they form a mat-like structure and they will reduce the soil erosion. The bryophytes are divided into liverworts and mosses. First, we will see about liverworts. So, liverworts will grow usually in moist and shady habitats. Like commonly, all the bryophytes will grow well in damp and humid condition. So, they are also a kind of this. And they can grow in marshy ground, damp soil, barks of tree, deep in the wood, and their plant body is thalloid. The example of liverwort is Marchantia. Thallus is dorsiventral, that is, it is like they are pressed. Okay, the leafy members have tiny leaf-like appendages in two roses, and the reproduction, the asexual reproduction takes place through fragmentation or by the formation of specialized structure called gemmae. These gemmae are green, multicellular, asexual buds which develop in small receptacles called gemma cup. So there will be gemma cups in Marchantia in which this asexual buds called gemmae are present. And when the gemmae become detached from the plant body, it will germinate to give new individual. And sexual reproduction is by production of gametes. So the sporophyte, that is a diploid sporophyte, it differentiated into foot, seta and capsule. The meiosis, because sporophyte is diploid, now we have to change into haploid to form the plant body. So the meiosis will take place in the capsule and they will produce spores that are haploid. This haploid spores will germinate to form the haploid plant body gametophyte. Next, mosses. And the predominant stage of the life cycle of mosses is gametophyte. Gamete producing plant body is only dominant here. This gametophyte has two stages. The first stage is protonema. This protonema directly developed from the spores, that is haploid spores, right? From that it will directly develop, that is protonema. It will be creeping, green, branched and frequently filamentous stage. In first lesson we have seen, right, fragmentation will take place in protonema of masses. So here it is. So protonema of masses are creeping, green and frequently filamentous. The second stage is leafy stage. This leafy stage developed from the secondary protonema as lateral bud. So here you see spores, from spores protonema, from protonema secondary protonema, from secondary protonema a lateral bud, from that lateral bud leafy stage. Okay, this consists of upright slender axis bearing spirally arranged leaf. Protonema was creeping but leafy stage was upright, and they have multicellular and branched rhizoids. What are rhizoids? Root like structure. And this stage bear sex organs. The reproduction is by fragmentation and budding in the secondary protonema. In the sexual reproduction, the male sex organ is antheridia and the female sex organ is archegonia. They produce gametes and it will form zygote. This zygote will develop into sporophyte, that is diploid sporophyte. The sporophyte in masses is more elaborate than the liverworts. Then it will form food, seta and capsule. Then capsule contains spore. This spores will be disposed and this spores will germinate the mosses. So examples of mosses are funaria, polytrichum and sphagnum. Here we will see the images. Okay. The first one, that is A and B, is Marchantia, that is liverworts. Here you can see gemma cup. This gemma cups will produce asexual buds called gemmae. These are rhizoids that are root-like structure. You see they are like drossemental. They are like pressed, right? So they have archegoniophore. So this is female. First one, A1 is female and second one has anthridiophore. So it is male. If you want to differentiate it, I have a simple trick. So here in archegoniophore, you see like a flower-like structure, right? They usually say girls are flowers. Okay, so you can remember like this one is girls and here you see some flat structure. So this one is male. And second one, this C is mosses, that is funaria. So the down one, gametophyte, that is haploid. It has rhizoid and main axis. This will give rise to sporophyte, that is diploid. It has foot, seta and capsule. This capsule will give rise to spores. From that spores, again the gametophyte will grow. Next one is sphagnum, that is a peat mass that is used for transshipment. We have seen, right? So this one, this is first here. The upper one is antheridial branch and the lower one is archegonial branch. See, it is like compact. So it is girls keep calm, like that you can keep. And this archegonial branch, first one is antheridial branch. Next we all move on to pteridophytes. Okay, we are here. Pteridophytes include horsetail and ferns, and these are grown for medicinal purpose and as soil binders. They can be used as ornamentals also. So this is the first plant type that has vascular tissue, that is xylem and phloem. Xylem and phloem or vascular tissue, like we have bloodstreams like blood vessels. These xylem and phloem will carry water and minerals and food throughout the plants. So pteridophytes are the first vascular tissue, very important. They grow in cool and damp places and they may also grow in sandy soil. And here you see the pteridophytes, the main plant body is sporophyte. Sporophyte is what? Spore producing diploid part of the plant. So sporophyte is main plant body. In bryophyte, the main plant body is gametophyte. In pteridophyte, the main plant body is sporophyte. This sporophyte has true roots, true stem and true leaf. Okay, and these organs possess well differentiated vascular tissue. The leaves, if you see leaves, they have true leaves, right? So these leaves are small, that is microphyllous in Selaginella, or large in macrophyllous in ferns. Okay, it is small in Selaginella and large in ferns. And now, sporophytes. Sporophyte have sporangia. Sporangia from where spores are produced. These sporangia are subtended by leaf-like appendages called sporophylls. In some cases, sporophylls they form a distinct compact structure called strobili or cone in case of Selaginella and equistem. You see, the sporophyte, there should be some place where meiosis will takes place and they will produce spores, right? In bryophytes we have capsule where meiosis takes place. Like that, in pteridophytes we have sporangia where meiosis will takes place. These sporangia are subtended that are surrounded by sporophylls. These sporophylls may, in some cases like in Selaginella and equistem, it will be closed to form a strobili or cone, and they produce spores. These spores germinate to give rise to inconspicuous, small but multicellular, free living, mostly photosynthetic thalloid gametophyte called prothallus. These gametophytes they require cool, damp, and shady place, and they need water for fertilization. Due to this restriction, pteridophytes are limited only to geographical region. Most of the thing we see in our immediate surroundings are angiosperms and gymnosperms. We don't see pteridophytes that much due to the requirement of water for fertilization, and they need a cool and damp place to grow on. And the male and female sex organ are called antheridia and archegonia, and they produce gametes that are carried by water, and when they fuse they form zygote. Zygote thereafter produce a multicellular, well differentiated sporophyte, because sporophyte are the main plant body in pteridophyte. In majority of pteridophytes, the sporophytes will produce spores, right? They are similar in kind. So that are known as homosporous. If they are similar, they are homosporous. But only in Selaginella and Salvinia, they produce two kinds of spores, that one is large, macro, and another one will be small, micro. That is known as heterosporous. Okay. Next, the development of zygote into young embryos takes place within the female gametophyte. In gametophyte, they will produce gamete, and that gamete will fuse to form zygote, right? The zygote will turn into embryo inside the gametophyte. So this phenomenon was the first step for the seed habitat. The seed contains the gamete, that is the zygote. The gametes will fuse, they will form zygote. Inside the seeds, the zygote will turn into embryo. So this was the precursor of seed habitat. There are four classes in pteridophytes. First one, Psilopsida. Example, Psilotum. Second one, Lycopsida. Example, Selaginella and Lycopodium. Third one, Spinopsida. Example, Equistem. And fourth one, Pteropsida. Example, Dryopetris, Petris and Adiantum. We will see the pictures. These are the examples of pteridophytes. First one is Selaginella. As you can see, they have true leaves, true stems and true roots. And second one is Equistem. Third one is Fern. And fourth one is Salvinia. Next we will move on to gymnosperms. Okay. Gymnosperms are like angiosperm, but their ovules are not enclosed by an ovary wall and they remain exposed. That is, the seed is not covered by a fruit. They are naked. Gymnosperm vary from small tree shrub to a large tallest tree. The tallest tree species of gymnosperm is the giant redwood tree Sequoia, and they generally have taproots. So here they have given a differentiation between Pinus and Cycas. So make a tabular column in the space to revise easily. So first we will see Pinus. So Pinus, they have roots that have fungal association in the form of mycorrhiza. So fungal association in the roots of Pinus. Whereas in Cycas we have roots called coralloid roots. These roots help in nitrogen fixation because they are associated with nitrogen fixing cyanobacteria. And the stems are unbranched in Cycas and it is branched in Pinus, and it is also branched in Sideris. The leaves may be simple or compound. The Cycas have pinnate leaves. Okay, and the gymnosperms have good ability to withstand all type of environment. In conifers, we see leaves in needle-like form. This needle-like form reduce the surface area, so, so that the transpiration will be less. And the thick cuticle and the stomata sunken, right? These two help in reducing the water losses. Next, the gymnosperms are heterosporous. We have seen that pteridophytes are homosporous except Selaginella and Salvinia. Here all the gymnosperms are heterosporous. They produce two types of spores. One is microspore, another one is megaspore. These spores are produced from sporangia that are formed on sporophylls. These sporophylls are arranged spirally to form a strobili or cone. The strobili bearing microsporophylls and microsporangia are called microsporangiate or male strobili. That is, if from a microsporangia we form microspore. Microspores is male gamete. Here, the male gametophytic generation. Gametophytic means what? The gamete producing generation is highly reduced and it is confined only to few cells. We see pollen grains, right? These pollen grains are reduced gametophyte. So the pollen grains are not gametes. They are gamete producing organism. So the pollen grains are reduced gametophyte from which the male gametes are produced. The cone bearing megasporophylls or ovules or megasporangia. That is, megasporangia is known as ovules. They are known as female strobilus. Okay, that is, they have male sex organ and female sex organ. Megasporangia, microsporangia. If this both the strobili or cones are present on the same tree, that is, they are bisexual, it is an example of pinus. So pinus is bisexual, whereas cycas, they have male cones and female cones on different trees and they are unisexual. And as you see, the megaspore mother cell will differentiate from nucellus. These nucellus are protected from envelopes. This ovule is megasporangia. These ovules are formed on megasporophylls which may be clustered to form female cone. Okay. One megaspore mother cell will divide mitotically and they form 4 megaspores. Out of that 4, one will differentiate to form female gametophyte. Female gametophyte will produce gametes. So that will bear 2 or more archegonia or female sex organ. That female gametophyte will have female sex organ. Unlike bryophytes and pteridophytes, the gymnosperms, the male and female gametophyte, that is gamete producing generation, they don't have an independent accession. They are retained within the sporophytes. And pollen grain. The pollen grain means male gametophyte is released from the microsporangium. This is carried by air currents. They does not require water. They require air currents and they will come in contact with ovule. What is ovule? Ovule is megasporangia. Once the pollen grain reach here, they releases the gametes, male gametes, and this male gametes will fuses with female gamete and they form zygote. This zygote will develop into embryo. Ovule, that is, the ovule is female megasporangia that will turn into seed which are not covered by the fruits. Okay, that's all. And we will see the pictures. First one is cycas and second one is pinus and third one is ginkgo. Okay, next we will move on to angiosperm. So about angiosperm or about flowering plants, you will see very well in class 12 chapter 2. Here they have given a glimpse. We will read it out. So unlike gymnosperms, the gymnosperms, they have seed that are not covered by fruits. Here they have seeds that are covered by fruits. And the pollen grains and ovules are developed in specialized structure called flowers. So flowering plants are angiosperm and the seeds are enclosed by fruits. Here also they may be from small tiny plant to a large tree. So a microscopic ulfia, that is smallest of angiosperm, to tall tree eucalyptus over 100 meters. And they are commercially useful and they are helpful for food, fodder and fuel. They have two classes, that is dicotyledons and monocotyledons. They are differentiated into two classes through the number of cotyledons they have in their seeds. The dicots are characterized by having two cotyledons in their seeds, but the monocots have only one cotyledon. And if you see the male sex organ, it is stamen, it has a filament, and an anther. The female sex organ is called pistil or the carpel. They have three things: the stigma, the stipule, and the ovary. The female gametophyte, அதாவது gamete producing generation, is embryo sac. The embryo sac is formed by meiosis. They have 7 cell and 8 nuclei. You see, they have 3 celled egg apparatus, 1 egg cell and 2 synergid, 3 antipodal cell and 2 polar nuclei. The polar nuclei eventually fuses to produce a diploid secondary nucleus. Here you will see very well. See. This is stamen, the male sex organ that has a filament and an anther. From the anther, the pollen grains are released. What is pollen grain? Pollen grain is male gametophyte, that are the carrier of male gamete. This is carpel or pistil, the female sex organ that has stigma, the landing platform for anther, style, and ovary. This ovary have ovules. Ovules are megasporangium. Okay. Sorry. Here from anther, microsporangium, they form microspores. These microspores are known as pollen grain. They carry the male gametes. Here you can see it. From megasporangium, they form embryo sac. This is the structure of embryo sac. You see, this is egg apparatus that has an egg and 2 synergid. The center one is central cell that have polar nuclei. And in the up, we have 3 antipodal cell. So you can see very well. So this is the structure of embryo sac. What is embryo sac? Female gametophyte. Pollen grain, male gametophyte. Embryo sac, female gametophyte. So from here, female gamete is produced that will fuse with male gamete and they will form zygote. Now as soon as the plant releases the pollen grain that carries the male gamete, that will land on the stigma. This stigma will allow the pollen tube to grow through stigma and style and they reaches the ovary. Inside the ovary is the embryo sac. The embryo sac has egg. Egg is the female gamete. So the pollen grain releases 2 gametes. One gamete will fuses with the egg and form zygote. The other gamete will fuse with diploid secondary nucleus to form triploid endosperm tissue. Okay. After that, the zygote develop into embryo and the triploid endosperm tissue develop into endosperm. Then they will give rise to sporophyte. Here they have given it. So the transfer of pollen grain from anther to stigma is known as pollination. 2 male gametes are discharged. One of the male gametes will fuse with the egg cell to form zygote and the other will fuse with diploid secondary nucleus to form triploid primary endosperm nucleus. This is known as double fertilization because there are 2 fertilization. And here zygote will develop into embryo and the primary endosperm nucleus will develop into endosperm. This endosperm will provide nourishment to the zygote. All these events will occur and after that the ovule will change into seed and the ovary will develop into fruit. In this diagram it is clearly explained. Next we will see the alternation of generation. So basically we will see a glimpse of it through this diagram. Gametophyte. What is a gametophyte? A gamete producing generation. So it will be N. So it will produce gametes through mitosis. Because it is N, it is going to produce N. So we need only mitosis. Now these 2 gametes will fuse to form zygote, which is 2N. So now we want to come back again to gametophyte, right? So we want to undergo meiosis, a reduction division through which 2N is converted into N, that is spores. Only this blue color part is the sporophyte and this one is gametophyte. These spores through mitosis, they will increase in number and they will form gametophyte. That's all. This is very simple. Gametophyte will be haploid and sporophyte will be diploid. Okay, so here you can see a clearly alteration of generation between gamete producing haploid gametophyte and spore producing diploid sporophyte. So this is known as alteration of generation. So these are different in different classes. First, the sporophytic generation is represented only by one celled zygote, that is sporophyte. The diploid condition is exhibited only by zygote. See, in the first, in option A, you see only the zygote is in 2N, other all are N. only zygote is diploid, and that is only sporophyte. So this type of alteration of generation is found in all type of algae, and this is known as haplontic life cycle. This is shown by Volvox, Spirogyra, and some species of Chlamydomonas. The dominant and photosynthetic phase here is gametophyte. Gametophyte is only more. Only sporophyte is only zygote. Okay. The second one is the extreme. The diploid sporophyte is dominant. It is all over. The gametophyte is not independent. It is dependent on the sporophyte. This type of life cycle is known as diplontic. An algae called fucus is also diplontic. And all angiosperms and gymnosperms are diplontic. You can see from option B. Here you see the gametophyte that will produce gamete. The gametes will fuse to form zygote, and the sporophyte is the dominant phase. It has multicellular and it occur for many times, and again it will undergo meiosis to form gametophyte. That gametophyte will produce gametes. That's all. They will fuse to form zygote. That is only of short time, and that is of only few cells. So gametophyte is not dependent. The gametophyte is dependent, and the sporophyte is the dominant and independent phase. And next come the intermediate, haplodiplontic. Here both the gametophyte and sporophyte both are multicellular. Okay, this is represented by bryophytes and pteridophytes. But the difference is the dominant phases. In bryophyte, the dominant and independent phase is the haploid producing gametophyte, and this is alternated with short-lived multicellular sporophyte, totally or partially dependent on the gametophyte. So gametophyte is only independent and sporophyte depend on the gametophyte, and the gametophyte is dominant means it is bryophyte. If it was vice versa, the diploid sporophyte is dominant. The sporophyte, the spore producing generation is dominant, and if it was independent, and if it alternates with a multicellular independent but short lived gametophyte, this is an example for pteridophytes. You can keep it very simple. Bryophytes, they have a dominant phase of gametophyte. Pteridophytes, they have a dominant phase of sporophyte. That's all. Interestingly, while most algal genera are haplontic, we have already seen algae are haplontic. There are few exceptions. Ectocarpus, Polysphonia, and kelps, they are haplodiplontic, and Fucus is an diplontic. Okay, this is very important. You should read the summary also. Okay, within one day I will upload the MCQs for lesson 3. You can go through it and get ready for lesson 4. Thanks for watching. Don't forget to like, comment and share. Stay connected and subscribe to Biology 360.